Fluorescent speckle microscopy is used to probe the dynamics of various cytoskeletal proteins in living cells. When used correctly, quantitative measurements of the velocity and turnover kinetics of cytoskeletal proteins can be made. We begin this procedure by microinjecting a small volume of fluorescently labeled actin into living cells. We then assemble the imaging chamber to reduce the effects of photobleaching. First, 2 pieces of double-sided tape are placed on both sides of the microslide where our cell cover slip will rest. Then, imaging media is pipetted into the chamber by dispensing the media near one of the open slits. Lastly, all 4 sides are sealed with Valep.
Hi, my name is James Lim in the laboratory of Gaudenz Danuser in the Department of Cell Biology at the Scripps Research Institute. Today we're going to be showing you a procedure on fluorescent speckle microscopy. We use this procedure to probe actin dynamics in migrating epithelial cells. So let's get started.
A key component to acquiring good fluorescent speckle microscopy movies is the proper labeling of actin with the fluorophore of your choice. We recommend using fluorophores of Alexa and X-rhodamine succinamidyl ester derivatives that target exposed lysine residues on the surface of actin. It is important for the labeling ratio to be high, around 0.3 to 0.7 dyes per monomer of actin, to ensure that speckles will appear bright and discreet.